Hello again from Dr. Kate. We're going to do a little bit of review here in what I like to call the logic of research. But first I'll show you yet another of the emails I get constantly from folks I've never heard of asking me to do some sort of a publication for them, sending them a manuscript, or in this case you can see they want a 1 to 2 page little editorial or something else. So I've never heard of whoever this guy is, but they always, when they send these, have a really tight deadline. So this is what, maybe a little over 2 weeks. And then I thought, well, what the heck? Let me see what this particular journal is about. So I Googled them, and here's the first thing that I see, that they want to charge me for the privilege of publishing in their journal. And that's the $2,700 is in pounds. And the way the exchange rate goes these days, that would be a pretty sizable sum to, again, just to say you can be published and add it to your curriculum vitae. So I passed on that fabulous offer, as I have so far with all of them. And maybe if I worked at a place that required multiple publications per year in order for you to keep your position, maybe I'd be singing a different tune. But for me here at our university, I'm going to pass. So again, this is the logic of research, and I want to reiterate and maybe restate some things in the hopes that this will be clear for you. So what's the first step? Asking the research question. We tried to do that with writing 5 pairs of questions related to some communication topic that was of your choice. Then the next step was for you to take one research question out of each pair and generate or craft the prediction, which is the hypothesis. What do we do after that? Based upon the question and hypothesis, we figure out what kind of a study we need to design, what kind of data or empirical evidence do we need to bring to the forefront in order to answer the question. And then we test the hypotheses via a computer program. The data that the computer spits out for us, one way or another, is then analyzed. And then we figure out, so did what we predicted actually happen or didn't it? And that's what we're talking about when we say we accept or reject the hypothesis. And then as we write up our research, the final part of the article or manuscript is to say, now what? This is what we found, and now we want to talk about how does that matter to what other researchers have found, and how does it matter to who we're studying. And we went through, in abstracting an article, the idea of internal validity. And what that means is the study itself good enough that reasonable conclusions can be drawn from it? And if you remember, for internal validity, there were the 3 questions that we always are asking when we abstract an article. And those 3 questions, if you can remember back, are number 1, were the independent variables properly identified and manipulated? And if you can look back on your notes for the 2 other questions of internal validity, that's always what we're talking about. And then we have external validity. And if you can remember that, relate it to generalizability. Is what you found applicable or generalizable to a population other than the population you studied? That's external validity. And when we do our research commonly in our field with college students, we really can't say that that has great external validity, even to other college students. And we definitely can't say how college students answered should also be applicable to all adults, even in the same geographic location, much less beyond, let's say, our university. Is what we found able to contribute somehow to a field? And a field of study has its findings which we are building upon. Those findings we call the body of knowledge. And that's really a goal for someone like myself as a researcher, that we're trying to contribute to and expand the body of knowledge. And also, is this going to really matter somehow to the quote unquote real world? Something we'd be interested in is, how can this help professionals do their jobs better? Maybe I want to research how to make customer service a better experience for our customers by offering some sort of training and seeing what type works best. That's what we would mean by, how can this help the real world setting? And I might find that one type of training, maybe an interactive online, is better than having someone face to face in more of a lecture setting. And that's a real contribution. So the goals of science are to explain, which we are good at, descriptive information in our field of communication; predict, which is when we create the hypotheses, and based upon past research, what do we expect to find? And ideally, we'd like to control the situation. And as I mentioned before, that's really pretty far out, if ever, to be found in a social science field, because we're talking about human behavior. And it's pretty unlikely we're going to be able to control everything so that we know with perfect predictive power how someone's going to behave. Now let's talk about the null hypothesis. You might have heard of the word null. What we mean by the null hypothesis is that instead of saying the harder as far as number of hours one spends studying a given subject in a week, the greater their comprehension of the material. That's a hypothesis. The null hypothesis would say there is no relationship between those two variables. There is no relationship between the amount of time one spends studying a particular topic and their retention of that material or comprehension. So the null is a statement of no relationship. Why is that important? Well, to be honest, in almost any circumstance you can imagine, most variables have nothing to do with one another. But what we do in the social science and in the scientific method is that we're trying to find relationships based on the literature. We go with what other people have found and try to build upon it. The null hypothesis is what the computer is testing for. What the computer is going to tell us is, based upon the analysis that is run, can we reject the null hypothesis in favor of accepting what we predicted. Now you see here in parentheses that in our world of social science, we have a threshold that we are comfortable with, and that threshold is 0.05 significance. P equals 0.05, and all that means is that, think of 0.05 as 5%. That if we, through our computer test, find significance at 0.05, we accept that hypothesis. That means 5 times out of 100, we're wrong. And that's the gamble that we are willing to take. And we always report these kinds of stats. We have to be sure, as sure as we can be, at least 0.05 for the probability level, or even lower would be nice. There's no such thing as a perfect probability that you're 100% accurate. There is no such thing. So all we do is, at best, accept the hypothesis. Now we have to do, as we're going to go through today, we're going to have to look at the data to make sure we understand what it is that we are either accepting or rejecting. Now I have a handout for this also in this week's materials. I'm going to go through them in slide form, but you might want to also print that out and take notes on it so that it's clearer to you. So you'll see it's a 2-page handout, which looks pretty much the same, each table, but there's a little twist with each one. So let's say the hypothesis is that having a female doctor leads to greater knowledge of illness by patients than having a male doctor. And I have just a little bit more information here that we had a 20-item true-false illness knowledge test that we gave to all of the patients. And I'll tell you that the 20 items, you would get 1 point for every correct answer. And further, that you would have a range of possible scores from 0, which is everything's wrong on the test, to 20, which means you have a perfect score. So here is what we're looking at. Male doctor, female doctor. The mean is the average, and we'll talk about simple statistics in next week's lectures. But for now, just think of mean as the average. We had 50 people, patients, with a male doctor, and after the male doctor explained what they were going to explain, the patients with the male doctor scored 15 out of 20 on average, whereas those patients that had the female doctor scored 16 out of 20. Is that higher? Yes. Does it matter? No. And that's the next line where it says T. That's a test, a t-test, which means you're comparing 15 versus 16. A t-test is one of the tests we call a comparison of means. So that number for the t is low, and we'll go look at the difference and how that relates to, can you see under the female doctor where it says P level, under the mean? P level equals 0.49. Well, we know that it has to be P equals 0.05 or lower to be statistically significant. So When I'm looking at data, the first thing I look at, honestly, is the P level. If it's greater than 0.05, that means the null hypothesis of no relationship between gender of doctor and knowledge of illness is supported, and we reject our research hypothesis of a difference by gender of doctor on knowledge of illness. So that's one example. Now let's look at another. In this example, again, we have the same hypothesis we're testing. And that is, in this case, we've got a mean for the male doctor of 15 out of 20 for the illness knowledge score by patients versus 17 for patients who saw the female doctor. You can see the T level is higher than it was in the previous example, but the P level is still above 0.05. And because of that, we accept a null hypothesis of no relationship and we reject our research hypothesis. It is not supported by the data. That's all it means. Sometimes when you read research articles, they will say it just like that, meaning that they will say the null hypothesis is accepted. And sometimes you won't see it. It depends on people's preference and style of writing. Now let's look at yet another example. And remember, the hypothesis is that female doctors lead to greater patient illness knowledge than male doctors. So here we go. Male doctor, average on the knowledge test, 15. Female doctor, average 18. And again, these are out of a possible 20 for a perfect score on this illness knowledge test. The T, which is the comparison of means, that's gone up to 2.58. How does that get actually calculated? I don't know. I haven't done it by hand. I mean, I know the logic of it, and that's what we're going for here, the logic of research. And the P level here is 0.03. And remember, we're looking for 0.05 or lower, so we've got it. And what does that mean? That means the null hypothesis of no relationship is rejected, and the research hypothesis of a relationship is supported. And we see here that the score on the knowledge test for females is significantly higher than the score for patients who had the male doctor. Take a look at this. In this example, we get the same T equals 2.58. We get the same P level. So we have to make sure we understand what we're looking for, because it's not just enough to say, oh, it's significant, just like we thought. Because look at the means. The computer is going to give you the exact same result. It's not going to know which direction that you are, in a way, rooting for the female doctors, for their patients to have higher scores, because that's what you were predicting. Nope, in this case, it's actually the male doctor's patients' illness knowledge test scores were significantly higher. So in this case, look at the two lines below. The null hypothesis is rejected because there is a relationship found, statistically significant, but it is not what we predicted. So the research hypothesis is not only not supported, but we found the opposite of what we predicted. And that would be an interesting thing to talk about. And as a researcher then, you try to figure out, well, what the heck happened there? And try to do some fast talking as far as what you write about your results. So let's talk about a few other terms. Randomization. This means every person who is part of your study, we call them subjects, sometimes we'll call them participants, respondents, kind of depends on what the author prefers as a term. But let's just go with subjects here. That if you do randomization, that means everybody has an equal chance of being in any condition or cell. Now think about an Excel file. When you open that up and you put numbers in the different cells, right? Those different boxes. So imagine here that you have 3 possibilities of where your research participants could take a look at, let's say for an experiment, 3 different styles or types or conditions is what we call them. Condition A, B, or C. And you get a group of good old college students to come in and do this experiment for you. As they come in, they are randomized to A, B, or C group. And the randomization is each person, regardless, has an equal chance of being in any of those groups. That's randomization. When you do that, and we're going to talk about this when we get to the experimental design, when you create that randomization, you are able then to say on a whole bunch of other variables, you are controlling them. Because if, let's say, you have intro to communication students, they're your folks who have agreed to be part of a study, and you put them as they enter the area where you're doing the experiment, and you put them as they come in, randomizing to room A or B or C. You then, if you do it correctly, and there's a process to it, if you do it correctly, then you can say in the end, if you find a difference between group A versus B versus C, and whatever it is you're measuring, you can say, all right, it's because I did the randomization process. The reason for it is, in all likelihood, because of the difference in the experimental rooms. Because everyone had a chance to see any of the 3 types, A, B, or C. And we'll return to that. It also helps with one of the questions of internal validity, the 3rd question, if you can recall that, which is that, did the researchers reasonably control for other factors? And that allows you to, if you've done it well, it allows you as somebody who is abstracting or critiquing an article to say, yeah, they did that properly. Now, just because you've done the randomization doesn't mean that our university's college students are like anything beyond our university. So because we picked a class that everyone has to take, Introduction to Communication, so that's fine. But can we say beyond the fact that these results, whatever they might be, can relate to university students at our location? We can't say it goes past that necessarily, beyond that, generalizable beyond that. And that's what we mean when we say the word universe. That is, if we say our universe are all American college students, We can't say just because we did something at our location that it's representative beyond that. So this design, as I mentioned, is what we use for the experimental type of method, the most tightly controlled, strictest method that we will start with shortly. Now with random sampling, and you might have heard of this term and maybe not known exactly what it means. This means that, let's say we want to do a sampling of all undergraduates at the university. And I have done this type of thing, but we're not going to ask every single person. All, however many there are, we'll say 12,000. We're not going to send out a request to 12,000 students. Instead, what we would do is work through the registrar and say, I would like a list of emails for randomly selected 20% of our undergraduate population. And that's who I'm going to sample. So this is very easily done these days with just the click of a few buttons to get that list, unlike what it used to be back in the day before we had the wonders of computer generated lists. So in a matter of moments, I'd get my 20% of 12,000. So if you do the math on that, hopefully you figure out how many that is. So can you do that? 12,000 is your starting point. I want 20%. So an easy way to do that is, well, for 10%, you would just take off one of the zeros, at the end, and then just double that. So we'd have our 2,400 we would send emails to. And then if it's done correctly, the random sampling means anyone had a chance to be part of that group. So the universe in this case was the entire undergraduate population, and then we randomly sampled 20%. And that then is a representative sample, and that's what we would be going for. This kind of process allows you to talk about the generalizability of your results, at least to that universe of your university's population. There would be external validity. And this kind of style is what we do in the survey methodology. So you might have heard of samplings of let's say adults likely to vote in an upcoming election. What you now as scholars, budding scholars of research, would be asking yourself, so how did they collect the data? Who did they ask? How was it done? You might notice that depending on what media outlets that you select, they might get different results for the same likelihood to vote for a given candidate. And it's because of the method, which is what we're talking about. And we'll get more into that as we move forward. So we're ready for the next step, and that is your first practice abstract. This is just a very brief, as you can see, when you take a look at it, it's only one page. It's not even a filled up full page, called The Research of Dr. K. The second page of that is what you, and again, print this out, what can you pull out of that page called the Research of Dr. K and transfer it over to that second page. So please take some time and do that. Don't panic. I'll let you know when it's time, if ever, to panic. And I've posted what the answers are, but it really would do you no good to just look at the answers and say, oh yeah, I knew that. What will do you good is to print out the Research of Dr. K, have your highlighters, whatever it is, method you like to use, circling what are the independent variables, what are the dependent variables, and then check yourself. And after you complete that, you can move on to the next PowerPoint. So I'm signing off for now. Good luck with your first practice abstract.